Welcome to Brain Ponderings. I'm your host, Mark Matson. Today it's my pleasure to talk with Eva Feldman. She's University of Michigan James Albers Distinguished University Professor and Russell DeLong Professor of Neurology at Michigan Medicine. So that's a mouthful, which means she's very accomplished in what she does. And she's also founder and director of Neural Network for Emerging Therapies. And her early interests were in peripheral neuropathies and ALS, I think. And then more recently, she's got interested in more the brain and Alzheimer's. But one thing that the main thing we're going to talk about is the so-called exposome, which is the total exposure you experience during your lifetime. So whatever, could be exposure to toxins or heavy metals. It could be simple things like temperature, I guess. Now we have climate change, and so people are experiencing a lot of hot weather. But the exposome isn't only exposure to bad things. It includes exposure to good things like exercise, not overeating, keeping your mind intellectually engaged. So welcome, Eva.
Well, thank you. Where to start, right?
Yeah. So before we went live, you indicated you grew up in Italy. Can you talk a little bit about your early life, what got you interested in science and the nervous system?
Oh, yeah, I'm certainly interested in medicine and science from a very early young age. I'm not sure why, because there were at the time, no other physicians in my family. I just always found it really fascinating, but decided upon medicine and particularly neuroscience when I went to a very small college in Indiana called Earlham College. So we moved from Italy. I actually insisted to my mother, who had us shuffling back and forth and who was Italian, that I wanted to go to high school in America so I could get into a good college. And so I went to this small liberal arts college, a Quaker college, and I had a neuroscience teacher and who I became close to. I was his teaching assistant, and he and his wife were just fabulous teachers. And so they got me so excited in the brain. So I knew then, geez, I knew what I was going to do. I was going to get a PhD in neuroscience. And then as I was doing my PhD in neuroscience, I thought, I better get an MD along with it. It's clear that I'd like to also be a neurologist. So that journey really just kind of slowly evolved over time, but very influenced by really excellent teachers and then really great mentors in medical school and then amazing mentors in residency. So my whole journey has been very influenced by all the individuals I've been very fortunate to meet.
And I guess you fell in love with the University of Michigan.
Well, yes, I did. I certainly did my MD PhD here. But then I went to Johns Hopkins and I really fell in love with Johns Hopkins too. I really did. I had amazing mentors there, Drs. Guy McCann, Jack Griffin, Dick Johnson, all names I know you know well, Mark, and just great mentors, great teachers. But I was married, and my husband wanted very much to return to Michigan. I thought, well, if there's any place I want to return to, of course, it's going to be Michigan. I've never regretted it for a day and have been really glad to be here. I mean, and I've been here now for 30, I have to do the math quickly, 35 years.
And looking back on your early work, so you were interested in a way very early on in what's now called the exposome, because you did some work on so people with long-standing diabetes can develop a lot of problems with neuropathies and their eyes, right? Retinopathy and
sure.
Yeah. So
so I think that, so I'll first say, in its broadest definition, you defined it well. The exposome is everything you are exposed to. So it's not only, as frequently people think, it's just environmental pollutants, air pollution, and everything from like sunblock to what you smell to what you can ingest. But it's really more than that, right? I mean, it is absolutely everything. So in terms of what you ingest, as you said, what you eat, how you exercise or do not, how you control your diet, and all in your lifestyle as a whole. And actually now we also know the social determinants of health actually are very important in the exposome. So with all of that in mind, when I was very young, I had started my clinical career in Michigan and noticed that I had a lot of patients with neuropathy, idiopathic neuropathy, in quotes. And what I noticed about these patients was that they were all overweight and none of them had frank diabetes. And so I began testing them for diabetes and found that almost all were pre-diabetic. So all of a sudden I thought, huh, no, they're not. And then the lore was you had to have diabetes to have neuropathy, right?
Yeah.
And that really began my journey in the complications of diabetes and pre-diabetes and obesity and the metabolic syndrome. Because really, the metabolic syndrome, obesity, hypertension, dyslipidemia, insulin resistance / diabetes. And when you think about the metabolic syndrome, now we know it causes neuropathy and it also causes cognitive changes. But many aspects of the metabolic syndrome can be altered or prevented if one looks at the exposome. If one actually looks at dietary practices, one looks at exercise practices, lifestyle practices, and then of course now we also know really you're influenced also by neighborhood, you're influenced by your zip code, your access to healthy food, your access to healthcare, and that's the exposome. So by chance, early in my career, I became interested in the exposome because in my mind, I was just focused on trying to convince the neurologic society, the neurologic community that you could have neuropathy without having frank diabetes. And that was really quite a heavy lift in my early career. Now of course it's well accepted as the field has grown and matured, of course. So the exposome really, diabetes, prediabetes, the metabolic syndrome, it's really part of the exposome. And then as my career, continues, and I have 3 NIH grants actually right now on, I guess you could call it the exposome and dementia now, because I've also become very interested in the metabolic syndrome and how it really adversely affects cognition and the mechanisms behind it. But as I'm going through this I would have to say this career, evolution, I really then became interested by chance, to a certain extent, on the exposome and Lou Gehrig's disease, or ALS. So would you like me to discuss that?
Well, I just like, one thing I'd like to say is that, since kind of the sequencing of the human genome and the ability to look at all your genes and their expression and so on, before that, there was some, not controversy, but just not knowing of what's the relative contributions of genetics and environment. And I think the diabetic problems with the nervous system is a good example where it may run in families not because of genetics, but because the kids are brought up in an environment where their parents have bad eating habits, don't exercise, and therefore they're more likely to follow those habits. But the genome thing, what it's clearly shown is environment plays a major role in most cases of Alzheimer's, Parkinson's, probably ALS, certainly neuropathy. So, and then the other thing maybe you can talk about is that advances big data and being able to analyze huge amounts of data, because when we talk about the exposome, obviously it's got to be huge, right? And in many cases, exposures are unknown. So can you talk about, like, how do you approach such a big problem of one determining?
Yeah, no, it's a
You're
great
never
question.
going to really establish the exposome for any one individual because there's always something you don't
Right.
consider.
So in order to answer that very broad question, I'd like to take it down to a specific disease, and that
Okay.
would ALS or Lou Gehrig's disease, because I can answer at each step of the way how we're trying to do it and how one would potentially approach it, or others could potentially approach the neurodegenerative disease. And what I'm discussing now has been similarly done by other colleagues, for example, in Parkinson's disease, and is ongoing in Alzheimer's disease. So first, when you talk about genetics, spot on, right? So we certainly, you can look at the gene, it's kind of the gene, time, environment hypothesis, right?
Yeah.
So you're born with a set of genetics. Over time, those genetics are modified by the environment through epigenetic changes, and which are mediated by the exposome. So you have this really close intersection with genetics and the exposome via aging and the environment. And we see that in neurodegenerative diseases. And that's one of the hypotheses, so you're spot on again, that underlies the genesis of multiple neurodegenerative diseases, including, I believe, ALS. That is our current hypothesis. So just to talk about genetics for a couple of seconds, of course, there have been multiple studies that show there's polygenic risk, right?
Yeah.
So it's not, a single gene mutation, but there's like 300 single nucleotide polymorphisms, and on these 300 individual genes that may be important, right? That confer on you a genetic risk. And so we developed a polygenic risk score for ALS, and there's clearly one for Alzheimer's disease and for Parkinson's disease, irrespective of the single gene mutations that can underlie all these brain diseases. So you have that polygenic risk. And then you can also then look at the methylome and the epigenetic changes and what's known as grim age. I love that term, right? So you have a methylome, you have so many DNA you have the set of DNA methylation events that occur in your lifetime. And there's a quote normal set. And if you've had too many, because you've had bad exposures, and this has really been shown to correlate high with air pollution, pesticides, pollutants, toxins, then you have excess DNA methylation. So, excess epigenetic changes, and those changes are then calculated. And when you have higher epigenetic changes that are outside your normal age bracket, then you have higher grim age. So the higher your grim age, the more likely you are to have neurodegenerative disorders. So genetics is so fascinating. And just like you said, too, the genetics with environment, parents who are overweight usually have children that are overweight. Yeah. Just as you've said. So, yeah, so there is that genetic component. But then the exposome and its intersection with the genetics. So, we have these circles. In fact, NINDS has now become very interested in the exposome, and they have what's called this One Tox program. And they have beautiful, really nice diagram where there's a big circle, the person's in the middle, and all the things that can affect the person, right? All the exposomic factors. So what we have done to understand at a level of, what is the contribution of the exposome to someone with a neurodegenerative disease, and in this case, ALS, we do the following. We do a very detailed occupational history to understand what are your occupational exposures. We measure in your blood legacy pesticides and pollutants. We look at where you have lived over the last 30 years to do geocoding. We're doing mass spec of blood and urine to look at unidentified toxins. These are just, we look at your diet, we look at your exercise profile. How close do you live to a lake? What kind of water do you drink? Is it well or is it from the tap? So it's very detailed. And with all of those, investigations, we've created what are known as environmental risk scores. So I could give you, if you want to come spend a couple of days in Michigan, your environmental risk score. And you are then scored by how many pollutants, your exposures, et cetera. And then we correlate, associate, your ERS, your environmental risk score, with your disease and with the severity of your disease. And we find certain big drivers of the environmental risk score, such as pesticides and legacy pesticides, which are everywhere, plasticizers and pollutants everywhere, exposure to metals, really ubiquitous. So we do that.
You know,
And
the
to
metal thing's interesting because, as you know, we have metals in our body that serve important functions. We'd be sick without them. Iron,
yeah,
selenium,
right.
but
Completely.
these metals are toxic at
Yeah,
high
well,
levels.
you know, an overdose, like anything. I was going to
Yeah,
say one last point, and then I'll come
yeah.
to metals. So in all these patients, we do a complete, so we completely do a complete transcriptome, we do a complete epigenome, we do a complete untargeted metabolomics, and we also look at inflammation. We do complete unbiased flow cytometry, looking at inflammation and all markers of inflammation,
And
or the
then,
inflammasome, and that's for each patient. So that's, you know, again, to try with this 30-page survey to get to the simple question you're asking, you know, what's in the exposome and how can we quantify it?
is the transcriptome done in the blood cells, the lymphocytes, or
Yeah, it's done from whole
from?
blood.
Okay.
We do have autopsies, but only from 100 participants. So we don't have a huge autopsy service for this particular disorder, but other groups, of course, are doing the transcriptome and autopsies. But you talk about metals, right? I mean, you know, welders have a much higher incidence and of course prevalence, but incidence of ALS
Huh.
than individuals who don't weld. People who work in the military, specifically with certain metals, much higher incidence of ALS. So, you know, certain occupations
Yeah.
really give you a really good idea of what are the clues in the exposome that are contributing to neurodegenerative disease.
So we know that lead and mercury can be really bad for the nervous system. What is it in the welders? Is it one of those, or is it some other metals?
No, it is, let's see, it's lead, it's zinc, I think,
Oh,
actually, and everything is in excess, and also the fumes. So I guess there are welder fumes. So the whole air pollution, or the whole thing, I know much more about air pollution now than I did 5 years ago, but I still have so much to learn. But there are a lot of, so polluted air is a really, even, and if it's, you know, occupational, of course, then it can also just be residential, then that's really a contributor, not only to ALS, or not only, but to, I would say, all neurodegenerative diseases.
wow. And okay, so you get all this data, and the idea is, and then you have like the clinical data on the individual, and I guess actually in some cases, ALS is much more rare than Alzheimer's or Parkinson's, but
Right. It'd be great to be doing this with Alzheimer's. We're not right now. We've got cohorts, obese, pre-diabetic, metabolic syndrome cohorts that we're following for cognition, but again, not to this detail. So this detail needs to be done. You made that point really earlier. What about population studies? What about big data? I mean, you know, what do we need to really drive the field forward? And I would just in some ways echo what you were implying, that we need big population-based studies, and when they need to be done well. I think, you know, what we've got, which are very good studies, is, for example, a retrospective look at insurance bases, databases. But
yeah.
what, you know, with ICD-9 codes, for example, so important studies. But I will tell you, a big group of, or a large new cohort that just completed, that I'm very excited about, although we don't have access to it because I don't have an appointment at our Veterans Administration Hospital, VA, but the 1 million veterans study is a very interesting cohort, Mark, and I think could answer many of the questions. Of course, it's skewed. It's 90% men, 10% women. And so that's a big, big red flag. But they have granular medical data on these individuals in the VA system, and you have open access to that data. Not if you need to be part of the VA system at this point to access it, is my understanding. They have biosamples, subsets are getting really deep cognitive phenotyping, MRIs, et cetera. So there's the kind of population you can follow them longitudinally and have a better understanding of what aspects of the exposome early on, like when you're 20, what is, you go out on binge drinking, for example. I mean, how is that affecting your brain when you're 60?
Yeah. Yeah. So that's in a way, so when you mentioned that, I thought of cancer, the old kind of idea, which I guess is true, that, cancer you have now probably began 20
Right.
years ago by, some mutation occurring in a cell or something like that.
Right.
And so it may be similar for neurodegenerative disorders, that some exposure you had early in life may predispose you later.
I'm sure. And, because of the DNA, I mean, again, for in terms of epigenetics and the methylome and, those changes now, as they accumulate, in particular, you get early ones now and they accumulate over time. I mean, it only makes sense, right? I mean, it
Yeah.
really, and we need to learn more in neurology from the cancer field. In terms of, they have done some really nice, larger than we have done yet, population studies,
Yeah.
which would be very important. And they're very interested in the environment too, of course, as you know.
Yeah. Now, people like to introduce, and I guess it's valuable, the term exposome, right? I mean, people have known for a long time that we're exposed to multiple things, and what we're exposed to may affect some
Right.
disease, but I guess coining a term like that can help.
Yeah. Now there's, right now there's, there's exposomic science is now a term, and there's whole exposome centers. We received an NIH Director's Transformative Grant based on the exposome in ALS. So it's very, not only is, I mean, so it's somewhat topical, but I think it's really, and again, I think you're implying this, it's just putting a name to work that we have all done in our
Yeah.
careers in one way or another, because we're looking at how external influences, however you define them, affect the onset and progression of disease and how they're interlinked with the pathology of disease. So we've all been exposomic scientists since day one, I feel.
Yeah, we did a lot of work a long time ago, right, in cultured neurons from hippocampus from rats, and we exposed them to iron. It causes free radical production and membrane lipid peroxidation and can kill the neuron.
Right.
And we just called that, when we published the study, this in the 90s, we just called it iron-induced neuronal death. We didn't call it theraptosis.
Yeah, that's
But
true.
then,
That's also a new field, which is interesting, isn't it?
yeah, I need to coin some term. I got one right now.
Oh, what's here?
It's the malexposome and the U-exposome. Malexposome is bad, mal is bad, and the EU, U is good. So malexposome and good.
You expose them. That hasn't been used as far as I know. I'm always afraid now these days, aren't you, to say that hasn't ever been done or that's never been said. I always say to my knowledge, because right, because who truly knows. I would like to discuss, if you want, and I'd like your opinion on this. I think so. We have all this accumulating data, and it's associative, but extremely strong. We have it for ALS. My gosh, the data for Parkinson's disease, Tim Greenemeyer's work is just mind-blowing in terms of the high association with Parkinson's disease and pesticide exposure. And then he's gone a step farther than we have, and he's shown it in rodent models. So his data is beautiful. We have all of these data. What would it take to have a public health campaign for prevention? I mean, we need to talk. I really like the idea of talking about prevention of neurodegenerative diseases.
Yeah.
I'm jealous of our Alzheimer's colleagues who really, I think, have done better than we have in, like, I'll say, the ALS field, and maybe even the Parkinson's field, talking about prevention of dementia and Alzheimer's disease. So what are your thoughts? Do you feel that the public is ready? You talk to all of us, right? Once a week, you talk to one of us. I mean, are we as a scientific community, are we as a lay public, are we ready to really have a big public health push for prevention of neurodegenerative diseases? Because, I don't see us curing them anytime soon.
Well, I think that is the message, and I think the public often gets the wrong message that there's going to be a treatment for Alzheimer's on the horizon, and there was this finding, and then the more accurate approach is say there are currently no treatments that slow the progression of these diseases, and we cannot be sure that there will be anytime soon. In fact, there's been a lot of failures. Drug companies have completely closed down their neurological disorder drug discovery, programs because of all these failures, and they're losing money. So I think it's almost like smoking. It's important we scare, or at least inform the people that we're all at risk. We're all at risk for Alzheimer's, Parkinson's as we age, and hopefully we do age into a ripe old age. But and the evidence suggests that, and this is why the exposome is so important, that what you do and what you're exposed to, your behaviors and the environment you live in throughout your life can affect and maybe determine whether or not you do develop these disorders before you die.
Completely.
So I think that has to be the message.
I do too. I don't think we're doing the best job of it. Because when you see a person with any neurodegenerative disease, what they want, of course, is the cure.
Yeah.
And I think, and my feeling as I've, getting longer in the tooth, been in this field a long time, is that we need to, as neurologists and neuroscientists, we need to really begin to think about prevention early on, like when people are in their 20s, let them know what the risk factors are. They make choices, but I feel that that's just not a message that's getting out to the general public at all.
And even parents, parents should know that the environment well, they do, but still, there's some bad eating habits.
There is.
And so we get the transgenerational kind of, I guess you'd say, propagation of chronic diseases because
there is. That's right. You're right. I mean, right. It is a transgenerational propagation of chronic diseases. That would be a name of a great review, Dr. Mattson. If you want to write a review for me for the Annals of Neurology.
Maybe.
I like that a lot.
Yeah, since I retired, I actually, I'm actually working on a third book now on
what?
eustress, EU stress, good stress.
Oh, well, stress is a big part of the exposome, of course. So what are you finding? Like what is
well, so the big three are exercise. When you exercise, it's a stress. Fasting, caloric restriction, that's a challenge. And then intellectual challenges. It's when you and I, what we're doing now, our neurons in our hippocampus and the connected cortical region, they're very active. That's a stress on them, just like when you exercise your muscle, free radicals go up.
Right.
But that's good because the cells respond adaptively. So
So like
anyway,
what's good stress, what's the right amount of stress versus overstress, right?
right. And medicine for a long time with people who had a heart attack, right? So long time ago, I think like I remember when in the 70s, even as recently as that someone who had a heart attack, their cardiologist would tell them, okay, you need to take it easy. Don't
Oh,
overdo it. You
all right.
don't put stress on your heart. But now we know it's important for you to actually exercise. Otherwise, your heart just gets weaker and weaker. So anyway, I think that there's a
Oh,
big role for physicians in understanding risk reduction
yes, me
under
too.
and then communicating that
To the
early on.
patient.
And I have a lot of problems with the healthcare system the way it is. It's all for
So,
profit.
right, you're in good company with most of us, right? I mean, in terms of access, time,
And money
reimbursement.
making, even at Hopkins, right? And I grew up in Rochester, Minnesota, Mayo Clinic's there, right? And it used to be when I grew up, and for a long time after that, all the physicians there were on salary. It didn't matter how many patients they saw, and they
Right.
would take a lot of time with individual patients, like an hour
Mm-hmm.
time. Now Mayo's profit driven, so they try to get essentially they're trying to run as many patients through the physicians as quickly as possible because they make more money for the institution. And I think that, it takes away from what a physician could do and kind of being a counselor and coach and
Yeah.
some and someone
Well, I
who,
think it's a real issue. I mean, I think um I don't think I know. Certainly academic medicine and medicine today is not what it was even 30 years ago in terms of resources, time, our ability to spend time with our patients, the amount of time we have for a new patient visit, because there's a lot of different and very real financial pressures on academic
yeah,
institutions. So it's like, how do we get that right balance? Because I want to spend time with my patients, and they hopefully want to spend time with me. But the times are now getting shorter for new visits, shorter for return visits, just as you said, as the academic institutions are responding to what are some really serious financial pressures. So what exactly is the end game? I don't think we know. I'm a proponent of, I guess you would have to say Medicare slash Medicaid for all.
yeah.
And that's not necessarily, I see that type of socialization of medicine as a positive thing, but it may not be doable, likely not doable in this country. And I certainly understand that, but it would be nice to come up with a happy medium, but I have no answers. I'm sure you don't either. And people who are way above our pay grade, who spend all their time thinking about the economic pressures, I don't think right now have answers. Things will change, though. They have to.
Well, you and I right now are doing some little bit, right? Hopefully that the people that are watching and listening will,
Prevention.
yeah, prevention. So
Right.
we still have 20 minutes or so.
Okay.
Let's talk about some specific things in air pollution. Oh, one thing we really haven't talked
Yeah.
about, you mentioned animal models. So
Mhm.
um, the exposome, the big data analyses like you're doing in ALS, where you get blood samples
Right.
and you analyze all these things and gene expression and have all the clinical data.
Right. Right.
Um, and some some specific chemicals that probably you're finding are linked maybe to a neurodegenerative disorder are chemicals that maybe were first shown in animal studies to have some sort of adverse effect on a health indicator. Um, and is the reverse true? It seems like to me that exposome, uh, scientists doing exposome and kind of the
Mhm.
association studies their work can inform
The animal studies.
the animal studies.
Of course they can. I couldn't agree with you more. We ourselves are not doing the animal studies. They're difficult to do. When you
Yeah.
think about it, if you're going to take rodents and begin to feed them, toxins, pollutants, pesticides, put them in polluted air. So they are difficult studies to do. So I have not ventured into that type of animal work, but work that we have done that I'm actually very proud of is we have looked at diet and exercise in rodent models and its effect on brain function, on cognition, as well as its effect on the peripheral nervous system. So we can take a mouse model and we can do one of many things, but we've particularly fed mice what is called a 60% high fat diet. So that's all of your that's 60% of your calories from lard, saturated fat, versus feeding a mouse 60% high fat diet from unsaturated fat, from essentially sunflower oil, oleate. And then we have exercised them, plus or minus those diets, reverse those diets, place them on a ketogenic diet, or also have them on a ketogenic diet only the entire time. So we've looked at the exposome in terms of diet and exercise in literally thousands of murine models at the murine, thousands of mice at this point. And what we find can be really summarized fairly easily. You can be equally fat, okay, equally chubby as a mouse, but if your fat comes from lard, you aren't very smart as a mouse. You have loss of cognition and bad peripheral neuropathy. If your fat comes from polyunsaturated, monounsaturated fats, you can be equally as chubby. You have normal cognition and normal peripheral nervous system function. So I think that's fascinating, right? I mean, so in my mind, a simple, not simple, but a really good public health movement on just diet could make such a difference in prevention in terms of dementia. We've also shown that if you lose weight, say you have a bariatric surgery, or which one can do on mice, or you really, go on a caloric restriction and you lose weight, you can have some regaining of your cognition. Pretty good, actually, and with a clear regaining of your peripheral nervous system, it seems to kind of get back in tune easier or better than your cognition. So a dietary reversal, you can actually clearly see improvements. Now, this is rodents. We are, though, seeing some of these same effects in our obese human cohorts where they've either undergone a very strict diet, supervised diet, where these shakes where you only get 1,000 calories a day, or bariatric surgery. And we also see a stabilization of neuropathy and improvement in cognition. So I think, so we've looked at that. And then we've also spent a lot of time, and I don't think it's for today's discussion, which you'll find it interesting in another time offline, the very basic mechanisms, like what's happening mechanistically. Yeah, like why does the brain not like saturated fats in terms of the beta oxidation cycle and what we're seeing, and ATP production, blah blah stuff you and I are very interested in, but maybe not for a general audience, versus why polyunsaturated fats just eat more, your brain's fine. And then there's the ketogenic diet, and then I'll tell you about that, and then I'll tell you about exercise. So the keto diet's interesting. The keto diet is not as effective as I thought it would be. I thought it would be just as effective as dietary reversal, as reversing a diet. And it's not as effective. The animals gain some cognitive ability, but you're much better off just losing weight by eating. So it's interesting because the ketogenic diet does have both saturated and unsaturated fats in it. Well, I'm talking about fully polyunsaturated, and it has very little protein. And the diet I'm talking about, the lard versus kind of sunflower diet, has 60% fat, but then also has 20% protein, and then 20% other carbs. The keto diet, remember, it's 90% fat and it's all saturated and unsaturated. It's got both and only like 10% protein. And so the brain doesn't really love that diet in terms of cognition in mice, mice. And then it does the peripheral nervous systems. And they lose weight just like people do. They're not so smart, which is interesting. And then my final point is exercise. Fabulous for all aspects of cognition. It is so interesting. And even these fat, because mice love to run on a running wheel. We did studies where we put mice on little treadmills, obese mice. They said, forget it. We're not going to walk on that treadmill. For you, there's little mouse treadmills, literally, envision it. We had 4 of them lined up. But now we give
so that they'll run on a running wheel, but they don't want to be forced to run.
Don't want to be forced to run, just like a teenager, as far as I'm concerned. So you put them on a run, but you give them running wheels in their cage and they'll run a lot. And we can access, how much they're running. Now, if they're on a lard diet, running isn't helping. But if they are any of the other diets, even the ketogenic diet, running clearly improves cognition. It's very impressive. It actually makes me exercise every day. My own experiments make me exercise every day.
Oh yeah, exercise has been a big part of my life. And then I had some accidents and couldn't exercise, and I got depressed. I think many people probably had this experience, but yeah, it's very good for mood, but it definitely improves cognitive function too.
Yeah, it really does. It's really quite impressive. I have a son who's a psychiatrist, and he's such a big believer in exercise, and he sends me all these studies, which really does show, exercise increases serotonin levels. I mean, it really does improve general mood. The exercise literature. I always say if I had to go back and do it again, I might become a kinesiologist. I think the exercise literature is fascinating.
Yeah. Well, it's definitely interesting, but
if you could do the
it's
basic
equally as interesting its effects on the brain, right?
Yeah, no, that's what I mean. There are studies. Have you seen those studies where exercise causes, this is a rodent models, exercise causes the birth of adult stem cells in the brain and the hippocampus. I mean, it's really impressive.
Yeah, so Henriette von Prague, she was under me at NIH, but I recruited her from Rusty Gage's lab at the Salk, where she showed that. And then when she came to NIA, she showed how these newly generated neurons integrate into the circuits, what.
And do they integrate? See, I haven't found this.
They definitely integrate. And the neurogenesis seems to play an important role in a particular type of cognition called spatial pattern separation, which has to do with navigation. So it kind of makes sense that if you we evolved.
Now this is going to be tangential, I think. I think I know where you're going. Tell me.
I guess what I want to say now was that I always think about our evolutionary history, right? And so we evolved moving through our environment, searching for food, avoiding hazard. And so our whole cognitive system is geared towards thinking while we're moving. And so I think one thing we really haven't talked about with the exposome is combining things, virtual reality, maybe you can do some things to affect your brain function and cognition, but it's not going to be the same as actually moving while you're thinking. You probably know this, if you go for a walk, you can think really clearly while you're walking.
Yeah, no, it's true.
I mean, I do this, I don't know if you do it when you write, but we do a lot of writing, and I'd write for a couple hours and then go for a walk.
Yeah.
And then while I'm walking, I'd think about what I've written, how it fits into the whole picture and so on. And then kind of write the next paragraph.
No, I agree. I mean, you think that's neurogenesis, or, I mean, why do you think that is? I think it's fascinating. It's true. I mean,
I
I'm
think you're probably getting more,
serotonin,
well, the
you know?
exercise literature, that there's peripheral factors, they're
Oh, yeah,
called
right.
exerkines,
Yes,
exerkines.
the extra kinds.
They're released and go to the brain, and one of them's been shown to increase BDNF levels, this neurotrophic factor that's so important for learning and memory.
Mm-hmm.
Um, we did some experiments where we combined actually treadmill running with
Uh-huh.
intermittent fasting.
Oh, we've done intermittent fasting too. I didn't tell you about that. But, these are in mice,
Yeah,
intermittent fasting in mice. And
yeah.
what did you find? What did you
Well,
find?
we were looking, the first thing we looked at is we trained them on the treadmill for 2 months. Every couple weeks, we'd increase the angle of the incline or the speed, so that they're increasing
Uh-huh.
their load. And then at the end of the 2 months, we did a maximum endurance test. And the animals that had been on intermittent fasting while they're doing the daily treadmill training had better endurance than the animals that were fed ad libitum, that is not on intermittent
That's
fasting.
interesting.
Yeah, and then we did metabolomics of the blood, and we
Uh-huh. Uh-huh.
took muscle and analyzed gene expression. And one thing we found was, well, this is known, exercise will increase the number of mitochondria,
Oh, yeah. Right.
right? And the intermittent fasting kind of boosted that.
Right. Oh, it did boost it.
It
Oh, that's
boosted that.
interesting.
And it also boosted the ketones that go up during the exercise. So you're getting kind of a bigger increase in the ketones that are a good energy source for neurons. And
Yeah, that's why I don't understand why the ketogenic diet, at least in our hands, isn't like a big home run.
I think the ketogenic diet, so you mentioned it has saturated, unsaturated. A lot of research now is, and this has been done in animals and humans by a guy, Steve Cunnane, up in Montreal.
Mm-hmm.
You give the animals or people medium chain triglycerides,
Yes.
which are immediate precursors.
Mm-hmm.
And also you can give them actually a ketone itself, beta hydroxybutyrate.
Butyrate, right.
Yeah. But okay.
Yeah, we're thinking about doing the medium chain triglycerides. I've got that on my kind of my list right now. What we're doing, which I think is fairly interesting, is this is an aside only for 2 minutes, and for any of the basic scientists listening, is that a very good investigator who's looked at temperature in animal rooms. Have you read this literature, and how that the temperature that's kept in animal rooms is too cold for the
Yeah.
animal, is they're not euthermic, and that actually affects our ability to measure true changes in cognition, true effects of diet, and true effects of exercise. So we're doing a couple of
Uh-oh.
look.
So
I
there's a lot of
know. I
people
mean,
publish all these papers and say, oh, maybe I should go back and do it at a warmer temperature.
well, we're doing just
Oh,
a real simple experiment at a warm, because now they've got a couple of rooms here at Michigan with warmer temperatures. So we're going to compare. Wouldn't that be so? And then once I do that, I'm going to do the medium chain triglycerides, because I think that's a really important piece of work to do. I really do.
another thing, the exposome I've thought about, I've got,
Uh-huh.
I have a vegetable garden, right? And it's like prime time. I'm bringing in stuff every day from the garden. And I've always been interested in it, and I think that the idea that fruits and vegetables have chemicals that sop up free radicals, and that's why they're good for you. That's not right.
I agree with you.
They do have some of those, but actually what they have is chemicals that induce stress response in our cells. They're actually natural pesticides. The reason they're in the plants is to keep
Oh,
us and other
wow.
organisms from eating them.
Really?
But
Is it?
but we co-evolved over, well, several 100,000. You go back even more than that, or
Mhm.
before. Um, we evolved to benefit from being able to tolerate these chemicals because we get energy and other nutrients from the plant. And so we have at least four things that keep us from overdosing. One is bitter taste. It turns
Mhm.
out, based on the science, that most of the chemicals that seem to be good for our health have a bitter taste. The second is vomiting. The third is there's enzymes in our liver that rapidly remove these chemicals, so they're only present transiently, as opposed to, say, man-made pesticides like DDT, which accumulate. The reason DDT accumulates is we didn't evolve with DDT around. We don't have any enzymes to get rid of it. But then the fourth mechanism that we've studied is direct effect of these chemicals on cells inducing adaptive stress response signaling, antioxidant defenses, and so on. So that's,
and it decreases the antioxidant defense, doesn't it?
yeah.
Yeah. I will tell you one thing. You mentioned DDT. So we have an enzyme that can take DDT to DDE, its first metabolite. It is ubiquitous in people and about 5-fold higher in people with Lou Gehrig's disease.
And does DDE have toxic effects too?
Well, I don't know, because I haven't done, I haven't done the animal experiments. I mean, that is the weakness of clinical, of human research. And then, you know, the ability then to take it back. In cell culture, yes, but
so then you'd predict
animals.
Was there a, can you look back in the history, was there like a blip in the increased incidence of ALS among people who had, because DDT was banned quite a while ago, right? So
right, but it's going to remain in the, you know, soil, water, environment for at least 100 years. So there is an increased incidence of ALS in farmers. And, you know, we have a beautiful farming bureau and cooperative throughout the United States. It's really quite impressive what the government does and how they keep very good statistics. There's a fairly high incidence of ALS in farmers, that's known. And although, you know, we have farmers in our cohort, it would take a very large study to, you know, to say is the exact effect of DDE, or, you know, how associated is it? See, that's the problem. These human studies are association studies, but I think there's enough. That's one of my ending comments. Don't you think there's enough association that we should just, like you say, get in there for prevention? I love your idea of writing a book on what eustress. That's right. And then maybe your fourth book, you know, proliferative friend of mine, maybe your fourth book could be on prevention. Let's prevent it all.
Yeah.
You got to finish the third one first.
My first one was on prevention, on the science intermittent fasting, but
yes,
I've been, for many decades, I kind of abandoned the idea that there's going to be some drug we're going to give to someone where they're already diagnosed and all of a sudden they're back to normal. It's not going to happen.
No.
You know, so
no. Not like insulin. I mean, you know, the brain isn't the pancreas. I'm always telling people. I so agree with you. I really do. Well, I'm going to ask you. I mean, it's your brain happenings, but I'm going to ask you a last question, if I could.
Okay.
And that is, no, I'm really very interested in your opinion. So, in the research you're doing for the eustress book, if you were someone like myself, and I see a lot of patients still, and I have brief amount of time to tell them, give them advice, but from your, you know, from the research, from what you've collected, what would be the one or two, maybe two things? I would have time to talk to them about one or two things that would be most important to maintain their cognitive health. What do you think it is?
Physical exercise and keeping your mind intellectually and socially engaged.
Okay, so engagement and exercise, the two E's.
And then moderation and energy intake, I think is.
Oh, you could do the 3 E's, energy intake. I
The,
like that.
the triumvirate.
Yeah.
Healthy brain aging triumvirate.
Another review you could write for me. Thank you so much for having me.
I've enjoyed talking to you, Eva, and keep up the good work. You've done a lot to help people. And
well, as have you,
you know,
sir.
if you can do anything to, you know, maybe if I can help to somehow impact enhancing getting the message out kind of at a high level, that would be great.
Yeah, thank you.
So,
And
so I don't know, like your annual meeting of the neurologists, right?
right.
Do they ever, talk, like have a working group on?
Well, I'm trying to start that on prevention,
Okay,
right? I
yeah,
really want to talk. I think we're definitely in agreement, and it's a, you're this esteemed basic neuroscientist, and I'm seeing, I'm a clinician scientist. I do the basic work, but I also do this clinical work, and it's where the two fields meet so well, I think, is in prevention. We both believe there isn't going to be one pill that's going to cure Alzheimer's disease or ALS.
right. Yeah.
Okay, thank you for having
Okay,
me.
but have a good
Okay.
rest of the summer.
Okay, you too. Bye, Mark.
Bye.
Bye.